Bear with me a little bit on this transport, and this should be a helping guide for you guys for 5.4 assignment about the transport. So there are two types of transport, the passive, that is here, and the active transport. The difference actually, for both of them, it's along the cell membrane, the movement of the particles. But we distinguish the passive and the active one. The passive is coming from a high concentrated area of that molecule to a low concentrated area. So it requires no energy. Many particles into a place where there's only very few particles. On the other hand, the active transport is when there are a lot of particles in here, and we want to just squeeze a couple of more from an area where there are very few particles. So it requires energy in the form of ATP, both of them along the cell membrane. Example for that, like the cell has inside 1, 2, 3, 4 calcium ions. The outside has 2 calcium ions. Now, let's analyze the pink arrow. If we have so many inside of the cells, but very few on the outside, and the calcium moves down its concentration gradient from the lot of calcium ions to a place where there's a few calcium ions, this is going to be a passive transport from high to low concentration area. On the other hand, let's check the blue arrow. And it says here outside is only 2 calcium ions, but inside there are 1, 2, 3, 4 already, and we want to squeeze that tiny calcium still inside of the cell. So it's against the concentration gradient, the movement of the calcium ion. So it's going to be an active transport from low to high concentration area along the cell membrane, and it requires energy. So it will be an active transport. The other one is the distinguish between endo and exocytosis. This is along the membrane when the molecule is super big. Usually it's polypeptides and proteins. Nothing else is going to be asked from you in the AP exam. So big molecules. Endocytosis means actually endo entering, getting into the cell. It's always the cell's perspective. Here is a protein outside of the cell, and the membrane opens in here, just start to engulf that particle. It can be receptor mediated. You will just check that in the video and in the textbook. But after that, when the protein particle is inside, enclosed in here in the cell, that membrane is going to pinch and binds off from the cell membrane. So the protein will be inside. It's a different story. It will just going to be used in a different way. That's okay. The exocytosis, basically exiting the cell by fusing the vesicle. The protein is enclosed in the vesicle by the Golgi apparatus. Think about the endoplasmic reticulum. RAF is making it, modifying it. The Golgi apparatus is going to packaging it. So it's going to send, the protein is going to be shipped to the cell membrane, where the two membranes, the vesicles membrane, the Golgi's vesicle membrane fuses with the cell membrane and let the protein or the big molecule leave the cell. That would help you guys with the 4.5, I guess, assignment. 5.4 assignment. Sorry about that. Okay, have a good one. Bye.